Welcome to the Learn Biology podcast, where we break down complex biological concepts and make them easy to understand. Today, we'll be continuing our series on cells. In this episode, we will be examining the cellular structures found in both prokaryotes and eukaryotes, and how complex cellular life evolved over time. To begin, it is important to remember that prokaryotes lack a nucleus and include bacteria and archaea, while eukaryotes have a true nucleus and membrane-bound organelles. Eukaryotes include single-celled organisms like amoebas and multicellular life like plants and animals. Now let's explore the cellular structures found in prokaryotes. The plasma membrane, a thin layer surrounding the cell, controls what substances enter and exit the cell and is composed of a phospholipid bilayer and embedded proteins. The cytoplasm, a gel-like substance that fills the cell, is where all cellular activities take place, such as metabolic reactions and protein synthesis. The nucleoid, a region in the cytoplasm, holds the DNA in prokaryotic cells. All prokaryotes have ribosomes, small structures made of ribosomal RNA and proteins, which play a crucial role in protein synthesis. Prokaryotes may also have a cell wall, which provides structural support and protection, and is composed of peptidoglycan in bacteria. Some prokaryotes have flagella, whip-like structures that allow movement, and a capsule, a thin layer of polysaccharides surrounding the cell wall that provides protection and helps the cell adhere to surfaces. Turning to eukaryotes, the defining feature is the nucleus, a membrane-bound structure containing the cell's genetic material, and the nucleolus, where ribosomes are assembled. The cytoplasm of eukaryotic cells also contains other structures, such as the endoplasmic reticulum, Golgi apparatus, and lysosomes. Eukaryotic cells also have ribosomes, which can be found in the cytoplasm and on the endoplasmic reticulum. The endomembrane system, composed of the rough endoplasmic reticulum, Golgi apparatus, transport vesicles, and lysosomes, processes and transports proteins within the cell. Lysosomes specifically break down food and cellular waste. Phagocytosis, a process where cells engulf and internalize particles or other cells, is important for cell eating and obtaining nutrients. Vacuoles are membrane-bound compartments that serve as storage areas for materials such as food, water, and waste. The mitochondria, known as the powerhouses of the cell, produce energy through cellular respiration. The mitochondria have an outer and inner membrane. The inner membrane has folds, called cristae, and are the site of ATP production. The cristae provide more surface area to allow for more ATP production. Mitochondria also have their own DNA and ribosomes. Chloroplasts, found in plant cells, are the site of photosynthesis. These organelles also have outer and inner membranes and their own DNA. The inner membranes are called the thylakoid and are the site where photosynthesis begins. So how exactly did cells evolve to have such complex organelles like mitochondria and chloroplasts? Endosymbiosis is the process by which one organism lives within another organism. Endo means inside, and symbiosis is a mutually beneficial relationship. In the case of eukaryotic cells, endosymbiosis is believed to have played a role in the evolution of mitochondria and chloroplasts. It is thought that mitochondria and chloroplasts originated from ancient bacteria that were engulfed by primitive eukaryotic cells and eventually evolved into organelles. In conclusion, cells contain various structures that work together to carry out the functions necessary for life. The differences in these structures between prokaryotes and eukaryotes reflect the evolution of complex cellular life. That's all for today's episode of the Learn Biology podcast. Don't forget to subscribe and leave a review. See you next time.